Hi there, everybody. My name is Dr. Chris Henstridge. I'm a Principal Investigator and Neuroscientist based at the University of Dundee. And I'm very pleased to present some of the work I've been doing recently in the lab. And the title of my talk is MND Connections. And hopefully you'll pick up the relevance of that title as we go through the presentation. So you may be wondering, who is this guy? And what I'm going to do to begin with is just give you a bit of an introduction to me as a person. So to try and sort of break those barriers of what the kind of general public often perceive as the quintessential scientist, the kind of Albert Einstein looking old white male. And hopefully you don't think I look like that. But to give you a little bit of background, I'm from up north, so literally the end of the line. If you take the train north while you're in Scotland, the last stop will be Thurso or potentially Wick, and that's where I come from. Although I'm a neuroscientist, I was never really destined to become a neuroscientist. My dad was a fisherman. Him and his two brothers owned a trawler boat and spent weeks at a time out at sea. And my mum, for a long time, worked in a nuclear power plant called Dounreay up on the far north coast. So I'm what you'd call, I guess, a first-generation STEM professional. So despite not having that kind of scientific background, let's say, I always had an interest in science. I always wanted to go to university. And I was always particularly interested in drugs and how drugs worked. And I always remember thinking when I was young, how a small aspirin pill, for example, could cure a headache. And so that little spark kind of led me to go to University of Glasgow to study pharmacology, how drugs work. And during that time, I spent a year working in Cambridge for a drug company called Celltech. And that really sparked and fueled my interests in research. And once I finished my degree, I came to the University of Dundee way back in 2005 to start a PhD. And that was really to start studying how drugs affected the brain in particular. So essentially moving into the field of neuropharmacology. And despite the picture, I actually really enjoyed my PhD, and I had really four and a bit fantastic years at the university. And when I finished my PhD, I moved to Budapest, so the capital of Hungary, joining the Hungarian Academy of Sciences. And that was an opportunity for me to learn new techniques and to expand my kind of technical background in studying the brain and how it ultimately changes in disease. After 4 years in Hungary, I moved back to Scotland, this time to the University of Edinburgh. And this is where my focus really shifted from models of disease into the actual human condition. And here, under the guidance of Professor Tara Spires-Jones, I started to work with human post-mortem material. So this was brains and spinal cords that had been donated by people who had lived with disease. And then a few years ago in 2019, I moved back to the University of Dundee, but this time to establish my own research lab and really start to focus on the research that interests me most. And the title that I kind of have for the research we're really focusing on at the moment is essentially understanding how changes at connection points between brain cells can cause disease. And you'll hear a little bit more about what this kind of really means and what we're focusing on as we go through some of the next few slides. So that's me, and I'm going to start talking a little bit now about the research. And at a motor neuron disease event, you might be a little surprised to see these three individuals on my slide. Doddy Weir, maybe not so much. But here we see three people who lived with three seemingly very different conditions. So Barbara Windsor passed away with Alzheimer's disease, Terry Jones passed away with frontotemporal dementia, and Doddy Weir, as I'm sure you're all aware, passed away a few years ago with motor neuron disease. And when you think about these conditions, you generally think about the main clinical symptom or presentation of these conditions. So in Alzheimer's disease, you think of changes in memory and thinking skills. Frontotemporal dementia, you maybe think changes in behavior. And with motor neuron disease, of course, it's primarily and generally considered to be a disease of movement. And if you look at brains that have been donated by people who have lived with these conditions, you will see changes in the brain in areas that you would expect based on the clinical presentation of those conditions. So in Alzheimer's disease, that red patch that I've highlighted is called the temporal lobe, and it's an area that's very important for memory. In the frontotemporal dementia image, this, as the kind of name suggests, is part of the frontal lobe of the brain and is important for planning and behavior. And then in the MND brain, I've highlighted there this thin strip, which is called the motor cortex. This is the primary location in the brain where the motor neurons sit in the brain. And the little strip at the bottom is the beginning of the spinal cord. And these are the areas that are primarily affected in these conditions. However, clinically, there's remarkable overlap between these three seemingly different conditions. So around half of people who live with motor neuron disease also present with some issues with their thinking skills and behavior. And a very similar number, between 40 and 50% of people who live with frontotemporal dementia, also develop changes and problems with their movement. And when you look at a large number of brains that have been donated by people who live with these conditions, you start to see that, in fact, there's quite a striking overlap in the areas of the brains that are affected as well. So you can see, for example, in the MND brain, we quite often see changes in the frontal lobe, and in people with frontotemporal dementia, we often see changes in the motor cortex and parts of the temporal lobe. So if we're aware that there's overlapping symptoms and similar changes in the brain, there's the potential here that if we study one of these conditions, we can dramatically increase our understanding of all of them and potentially develop ways to treat all of them rather than one condition specifically. And one of those overlapping changes is a very early loss of synapses or connection points between brain cells. The image at the top here shows you an example of what a human brain looks like. And if we zoom in really closely, we can see the individual neurons or the brain cells that make up the brain. And then the connection points, so essentially where these two brain cells communicate, is called a synapse. And we know that in the very early stages of conditions like Alzheimer's, frontotemporal dementia, and motor neuron disease, that synapses are lost often many years before the actual neuron itself dies. So if we can figure out processes or the mechanisms that are driving that synaptic breakdown, we may be able to stop it and stop these diseases at their very earliest stages. So I've talked a lot about these synapses or connection points. So what are they? Well, as I say, they're the connection points between brain cells. Amazingly, we have 10 to the power of 14. So that's 100 trillion of these connection points in our brain. And the brain cells never actually physically touch. They come very, very close together, and a message is transferred from one cell to the next at the synapse. And it's that transfer of information that controls the function of our brain and all of the outputs from that. So all of our memories, all of our dreams, our thoughts, our plans and movement are all controlled by this transfer of information between cells. Incredibly, we have more of these connection points in our brain than we have stars in our galaxy. And to try and put that in some context, if we could count 1,000 of these synapses per second, it would still take us almost 32,000 years to count them all. So obviously it makes sense to have, if we have so many of these in our brain, they must be very, very small. And in fact, these synaptic connections are thousands of times smaller than the thickness of a single human hair. But despite their tiny size, they're absolutely essential and fundamental to our very existence. They're the building blocks of our learning processes, our memory, our thinking, and our movements. They're also very dynamic processes or structures. So when we learn a new skill, for example, how to play the piano or how to learn a language, it's the constant repetition and practice of those new things that starts to formulate and build those synaptic connections between neurons. But if we then stop playing the piano or stop learning the language, the lack of that repetition and use of those networks means that they start to break down and are slowly removed because we no longer need them. And when we talk about the synapses or the connection points within the motor system, we talk essentially about the connections between the brain cells that sit in the brain in the motor cortex. And they connect with each other and they regulate the activity within the brain, but they also send long projections down into the spinal cord where they then connect with what's called the lower motor neurons, which sit within the spinal cord and project out into the muscle to control muscle contraction. So these are the main points of connection to the motor system, and we're studying how these change in the lab, and we'll talk a little bit about that, about how we do that. So we use two main approaches for studying synapses. We can use microscopes to look at synapses in donated human tissue, and we can also physically remove synapses from donated human tissue and see how they've changed in disease. And the technique that we use for that is called proteomics, and I'll explain a little bit more about what that means in a second. So using these types of approaches, what have we discovered? Well, just by purely counting the number of synapses, we've made some quite interesting observations in people with ALS, the most common form of motor neuron disease. In this study in particular, we took part of the frontal cortex, part of that frontal lobe that I showed you in some of the earlier slides. It's an area that's known to be affected in some people with ALS, and it's a major site of change in people with frontotemporal dementia. And by using our imaging techniques, we could count the number of synapses that were in the ALS group compared to a non-disease group. And we found a small but significant decrease in the number of connection points in the brain. But importantly, the people who were in this study, before they passed away, they undertook a screening test called the Edinburgh Cognitive and Behavioural ALS screen, or the ECAS screen. And that's a screen that was set up in Edinburgh that allows us to understand how well these people with ALS still think and behave. And from our 29 MND cases that we analysed, 23 of them have been cognitively tested with this ECAS test, and 7 of them were shown to be impaired. So that's 7 people with ALS and a change in their thinking skills and behavior. And now when we split the ALS group by the presence or absence of that cognitive impairment, we find those that have ALS plus cognitive impairment have the lowest synapse number. So this was the first time it had been shown that a change in the number of these connection points associated with a change in thinking skills in people with ALS. Now, the other approach that I mentioned was proteomics, and that allows us to profile the content of the synapse. Now, the image that I've shown you here is not a particularly good one, but it's from one of my review articles that kind of summarizes some of the key proteins found at the synapse. And you can see just from this small number of proteins in this image that synapses are very complex, and in fact they contain many thousands of proteins, so they're infinitely more complex than this image that I showed you here. So what I want you to try and do is think of a normal, healthy synapse as a Marks and Spencer's fruit basket, and think of the basket itself as the membrane holding that synapse, and all of the contents representing the proteins within it. And now consider the ALS synapse as a Marks and Spencer's luxury fruit basket. And what we can do is from these fruit baskets, let's say, is compare the contents and see how they change in a control synapse and an ALS synapse. And essentially the proteomics approach allows us to do an unbiased screen of all of the contents, so all of the synaptic proteins. So if we just list what's in the Marks and Spencer's fruit basket and the luxury fruit basket, you can see straight away there's differences. You can see there's differences in the number of items and the types of items. You can see we've got 12 items in the control synapse, or the MNS fruit basket, and we've got 21 items in the luxury fruit basket. But in fact, if you're actually looking at control synapses and ALS synapses, we've identified more than 6,000 proteins combined. Looking at the fruit baskets again, we can see changes in the contents. So here you can see the ones that are highlighted in red are new items that have appeared in the luxury fruit basket that we don't see in the regular one. So satsumas, a melon, and physalis are in the luxury fruit basket, but not in the regular one. The green ones show a quantity change. So plums are found in both of these fruit baskets, but you can see there's more in the luxury fruit basket than the control one. And interestingly, the blue highlighted item here shows a different subtype. So they both contain pineapples, but the luxury fruit basket has a golden pineapple. And these types of comparisons and observations we can do with the proteins that we find in the synapses to see how they change in ALS compared to control. And we found hundreds of different proteins have changed in the ALS synapse, both in terms of identification, so new proteins appearing, but also a change in the number in the ALS synapse compared to control. And this approach, I think, is really important because it gives us a much more complete understanding of the changes that are occurring at the ALS synapse. And hopefully, what I think it's also showing us is that it's revealing hundreds of potentially new drug targets that we could use as ways to try and treat the conditions. So that's what we've been doing. What are we planning to do now? So essentially, we're going to build on that. So most of the work we're doing is really still trying to characterize the human donated brain and spinal cord material in as great detail as possible, so we can get a much more complete understanding of what the human condition looks like. Based on all of that information, we will then refine and improve model systems to make sure that we're really testing the drugs in the most appropriate model system. So we're going to be generating models using human cells in a dish, often referred to as iPSCs. We will also use fruit fly models of different diseases and then start to change components of those fruit flies to see how that affects the disease onset and progression. And we'll also be working with some mouse models as well to test some of our ideas that come up from the human analysis work that we're doing. And importantly, the work that we develop in the model systems will inform what we look for and test for in the human material and vice versa. And ultimately, by combining all of this analysis, we hope to understand and eventually target the very earliest steps of human disease. And we're really trying to tackle these three, I think, very basic but fundamental questions. So where, when, and why do we lose synapses in disease, and how can we stop it? And finally, although I have given this presentation today and we've talked a little bit about some of the work that we were doing, it's always essentially the royal we. So as a lab head, I'm very fortunate to work with some fantastic colleagues. And at the moment, we have two postdocs, Dr. Zsófi László and Dr. Dinya van der Hoorn, working in the lab. We've got a couple of PhD students that recently finished and have since gone on to have their own postdoc careers in different places. We work very closely with colleagues across Scotland, the UK, Europe, and even now much more internationally as well. And we're very lucky to be funded by a whole host of both local and national charities and funding bodies. And of course, most importantly, none of this work would have been possible at all without the remarkably generous donation of tissue from people who have been directly affected by these conditions. So I would like to extend my thanks to everybody on this slide and beyond. Thank you.